draw the spindle fibers, all the stuff. Don't need. We're just going to deal with chromosomes today only. Okay? Mitosis, prophase. Okay, ready? Alright. Javier, will you come up to represent mitosis, prophase? Thank you. Ah, it's split out in two. Okay, as Javier is rearranging, everyone think up. What's the haploid number of chromosomes? Just based off this number of chromosomes here. Right? What would be the haploid number of chromosomes if this cell went through meiosis? Again, what would be the haploid number of chromosomes based on the number of chromosomes you see on the whiteboard? What would be the haploid number? Okay, maybe if I tell you right now, maybe you tell me how many chromosomes do you see on the whiteboard? 6 chromosomes? 6 chromosomes. Let's say that's the diploid number. Okay, the diploid number. Often we represent diploid by 2n. N represents one set, how many chromosomes there are. So what's the haploid number of chromosomes? It will be just 3 chromosomes. So more questions. Let's talk about homologous chromosomes. That's something new that you encountered over the holidays. So, how many pairs of homologous chromosomes do you see here? 3 pairs of homologous chromosomes. Okay. What makes homologous chromosomes homologous to another chromosome? Okay. What? I'm just going to choose these 2 chromosomes right now. These 2 chromosomes are homologous to each other. But what makes them homologous to each other and not, say, these 2? Why not these 2? Why these 2? What makes them homologous to each other? Okay, feel free to discuss. Alright, so what makes these 2 chromosomes homologous to each other? Okay? The word homo means same. So what makes them homologous to each other? Weird?
They have the same gene, no chi.
Okay? They have the same gene, no chi. Yeah, it's pronounced as loci.
Oh.
Not gene, no chi. You need to know this. Now then you go to university, then you tell everyone on the first day of school, this is the same gene, no chi. Okay, gene loci. What does gene loci mean to you in simple terms?
The same letter.
Same letter, okay? Same letter. What do letters mean?
Is it like the instructions for certain characteristics?
Okay, we are saying same instructions for certain characteristics. Okay. Actually, the word loci relates to location. Okay? The word loci relates to location. So we say homologous chromosomes have the same gene loci, we simply mean they contain the same genes in the same locations. Okay? if I were to, remember these are condensed chromosomes. If I were to untangle this, right, untangle it out, and then we zoom in a little bit more, you'll start to see all the A, T, G, C, G, C, so on and so forth. Maybe T, big, yeah, and so on and so forth, right? If we untangle it, actually it's just DNA. Okay? Last year, we learned that, I try to sneak it in at least, okay? Your DNA contains information, and the information can help your ribosomes decide what kind of proteins are made. So let's say this segment of DNA results in a particular protein to be produced. Okay? We will learn a little bit more about this arrow in one whole chapter. Okay? We call this segment of DNA that codes for a protein, a gene. Okay? A gene is just a set of instructions. It's a recipe book. Right? So our DNA is just an extended recipe book containing a lot of recipes of how to make this protein, that protein. Somewhere along this chromosome, there is a gene that codes for the protein that determines eye color. Right? Somewhere else along the chromosome, you may find a gene that codes for the protein that determines your hair color. Right? Somewhere along the way, there may be another protein that codes, another DNA, a segment of DNA, a gene that codes for protein, maybe an enzyme that determines how you digest food. Right? Proteins are very powerful. They can be structural, they can be for chemical reactions, many processes. As you were doing the SLS, you realize that system chromatics have the same, exactly the same thing on the left and right. So you see A, B, C. Right? So what do these alphabets mean? The alphabets are just a representation that this segment of the chromosome contains a gene. Okay? This segment of the chromosome contains a gene, and we just represent it in alphabet. Sometimes you will see that at the same gene location or gene loci, it's okay. Have a quick end. Eat a bit first. Give me a while. Okay, maybe while waiting for me, I'd like you to think about what's the difference between a gene and an allele. This is the next thing that was encountered during your holiday. Okay, gene versus allele. What's the difference? Okay, what is an allele? Okay, to put in simple terms, an allele is just a different form of the same gene. What in the world does that mean? Okay? An allele is a different form of the same gene. Sometimes we represent different forms of the same gene by same alphabet but just small caps. Okay, same alphabet but just small caps. Different form of the same gene. Okay, we represent a gene with an alphabet. But different alleles, we use either a small, well, what's the opposite of caps? Small caps, okay. Either small caps or big caps. Okay. Why do we have different forms of the same gene? Well, think about it. All of us have hair. Right? But some of our hair brown, some gold, some black, right? We all have the gene for hair color, but we can have different forms of the gene, right? Some of us may have the allele, maybe small a represents brown hair, maybe big A represents black hair, right? So alleles are just different forms of the same gene. A gene codes for a characteristic. Characteristic. It looks wrong. Okay, a gene codes for characteristic. Allele codes for a different form of the same characteristic. So for example, this big B over here will represent an allele that codes for blue eye color. This one, the small b, codes for black eye color. Right? It's different forms of the same gene. The next, a few chapters down the road, we will learn how having different combination of these alleles affects what we see on the surface. Right? What if this person has 2 copies of big B? How would that affect you on the surface, right? Characteristic wise. What if a person has small b, 2 copies of small b? How would that affect the person on the characteristic level? Right? We'll learn that in subsequent chapters. Right? But for now, all you need to know is what's the difference between allele and a gene. Allele is just different forms of the same gene. Okay? There are many genes on a chromosome. So A, B, C. Right? Often we just give you maybe K. But if I were to look across the entire chromosome, there could be thousands of genes, thousands of alleles. Right? But we often don't talk about that. Right? Often we just maybe pluck 3 from thin air. Okay? Okay, so that's what makes homologous chromosomes homologous chromosomes. Okay? A misconception is that homologous chromosomes need to have exactly the same allele. Okay? So I'm going to write it out here. Homologous chromosomes do not need to have the same alleles, but they have the same genes. Okay? Take a while to digest what I just wrote. They do not need to have the same alleles, but they need to have the same gene. Okay? Maybe more specific gene loci, then it sounds a bit better. Okay, same gene loci. Okay? So what makes them the same is that, yes, we have a gene that codes for eye color, but I can different forms of it. I have a gene also that codes for hair color, but I can different forms of it. Okay? So homologous chromosomes are not necessarily identical. They are not identical at all. Yes. Yeah, correct. Yeah, or as Sandra put it succinctly, they basically must have the same alphabets. Okay, but can be big or small camps, doesn't matter. Okay, but must have the same alphabets. The alphabets represent the gene, but whether it's small cat or big cat represents the form of it in any case. Okay, so let's plug this back in. Oh yeah. Now I'm going to change the alleles up a bit. Okay, let's go to after prophase is metaphase. Alright, mitosis metaphase. So, mitosis metaphase. Will you rearrange it? Oh, shucks, not long enough. Oh, what you want to do? Okay, you want to put everyone in one go. No, okay, we squeeze that. Okay, let's make the cell of this one. Still no space. Yeah. Okay. So metaphase. They're lined up in a single palm. Okay. Okay. So that's metaphase. Let's go to anaphase. Even no space. Okay, anaphase. Anaphase, I'll do it. I think the more interesting part is how things are different in meiosis. Okay, let's go anaphase. Anaphase, they will speed up, right? Yeah. By right, it will go. It will pull centromere first, right? Kind of like that will be more accurate. Okay, but if I flip it around like that, it's not accurate. Things turn along the way. Okay. So let's imagine a plane that you have got apart. Hello phase, it will jumble back together. Okay, and then after that become two cells, right? Then let's say this goes through S phase. Okay, this goes through S phase. Okay, so again, if this goes through S phase, what will happen? Okay, we ignore this fella, maybe this fella went on his merry way and enjoys life already. Okay, so we look at this cell again. This goes through S phase, it goes through S phase, you'll get another copy, right? Attached at the centromere. Goes through S phase, you get another copy attached at the centromere, attached at the centromere, attached at the centromere. So we are back to square one, but now let's talk about meiosis. How is meiosis any different from the process of mitosis? Okay, so now it takes some time to think about. What would it look like? Meiosis prophase. Okay, what would things look like in meiosis prophase using these same chromosomes? Right? Feel free to talk to your friends. Okay, what would things look like for meiosis prophase 1? What would things look like meiosis prophase 1? Okay, ready? Meiosis prophase 1. What would that look like? Scroll in. Suicide crystals. Okay, what will it look like meiosis prophase 1? Okay. Oh. Okay, so Colin has just positioned everything, and this is what she thinks it looks like meiosis prophase 1. Okay? Now I'm going to write some of these terms on the whiteboard. You try to describe to your friend how you use these terms in a sentence. Alright? The words are synapses, bivalent, chiasma, crossing over. How will you use these terms in a sentence to describe what you see here? Or chiasmata. Right? Without looking at textbook, you use these words to describe your friends what's happening on whiteboard. Okay, alright, so it's like English class, right? Okay, so I'd like to invite someone. We try to use these four terms in a sentence. Now, use text word, let's try. Okay, some are verbs, some are nouns. Which is which, so I've not thought about it yet. Ready, yeah? Okay, let's try to use this in a sentence. Talk to you.
Synapses is the process where homologous chromosomes come together form the bivalent.
Okay, synapses is a process where
The homologous chromosomes form the bivalent.
synapses is a process where homologous chromosomes come together to form a bivalent. Okay, two things check. Tobias right. Synapses is a process. Process 2C1C. Okay. Okay. Any more?
Crossing over
Crossing over occurs at the chiasmata. Okay. All right. Any other variation of how you might use these terms? Tell me now. Yeah. Yeah. Okay. Chiasmata is the site where crossing over takes place. Okay? I think that's fine. Right? If we put things in simple terms, if I did not use the word synapses or chiasmata, I could still describe this. I could say that homologous chromosomes pair up to form bivalents, and there are points where they cross over. Okay? Then we add in all the more complicated words. Rather than saying homologous chromosomes pair up, I say synapses occurs. Right? So synapses is just another way of saying homologous chromosomes pair up. Okay? So synapses just means pairing up. In simple terms. So I could say in meiosis prophase 1, synapses occurs. Right? Can or not? Right? Synapsis is a process. Synapsis means that homologous chromosomes pair up. I could say that meiosis prophase 1, synapsis occurs. Right? Because synapsis is a process. During synapsis, right, the homologous chromosomes come together to form bivalents. Okay, so what's a bivalent? Bivalent just means two chromosomes together. Bi, that's why there are two. You know what valent is? Okay. Chiasma or chiasmata just refers to the point where they they overlap each other. Okay, this point over here is a chiasma. Chiasmata is the plural of it. Plural means got a lot. Okay, so here is another chiasma. Here is another chiasma. We see 3 chiasmatas. Could a homologous chromosome have many more chiasma? And so here they overlap 2 times. So chiasmata. Homologous chromosomes can overlap several times along the whole way. Okay, so that's how we will use these 4 terms. Synapsis occurs. During synapsis, homologous chromosomes pair up, form bivalents. At the chiasmata or chiasma, crossing over occurs. What happens during crossing over? Okay, what used to be this? Big B, small b, right? When they cross over, what they do is that they do a swap. They break apart. This segment over here will break off and transfer to the other side. This segment here break off and transfer to the other side, such that I swap around the alleles. Okay, there's an exchange of alleles. That's what crossing over means, that we just exchange alleles. I have a question for you. Why is it that, let's say, C, D, 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 C, C. Why are the alleles of sister chromatids the same? Okay. Why are the alleles big C, small d, big C, small d, small c, big D, small c, big D? Why are the alleles the same in sister chromatids? Why are they the same? Why don't you find sister chromatids existing in this form at the start? Notice I kept drawing same thing on both sister chromatids, right? Why? Why did I draw it that way? Why are they identical? This is crucial. And why are they identical? Okay. Why are they identical? Because they just came from a phase called S phase. In S phase, all these chromosomes were but one by themselves. After DNA replication, we create an identical copy and attach them at the centromere. Okay? But during prophase 1, they can overlap with each other, and they could do a little swap around if you want to. Okay, we could do a little swap. So that means after prophase 1, sister chromatids need not be genetically identical anymore. It can be different. Okay, because we've done a little shuffle of the cards. That's actually what we're doing with ourselves. Every time we produce a sperm or egg, we just shuffle the cards before we redistribute it. Right? Else, every child that comes out from 2 parents, right, will look exactly the same each time. Right? Okay? Okay, so one last thing. Okay, so the idea of what is sister chromatic versus non-sister chromatic. Okay, I saw in your quiz, a lot of you wrote they form bivalents, they form chiasma. So that's crossing over. A lot of you wrote between non-sister chromatids. I wonder if you understand what that means when you write it down. Or you just lift it from textbook. So what does it mean? Crossing over takes place between non-sister chromatids. These two are sisters. Okay, so cis. These two also sisters. Right? However, if I look at these two, they are non-sisters. Okay? They are not sisters. Sisters are sisters if they share the same centromere. They share the same home. Okay? If you don't share the same centromere, they are not sisters. So crossing over takes place between non-sister chromatids, like that. Okay? Between non-sister chromatids. What if crossing over took place between sister chromatids? What would that look like?
The same.
Still the same? Can it take place? It can. Okay? Crossing over can take place between sister chromatids, but it's pointless. Right? If I were to cross over this over here, I don't know how to cross over, maybe like maybe they break off and then they soak apart. Still the same thing. Yeah? Okay, it's like you own self shuffle yourself and bad energy. Okay, so you don't cross over between sister chromatids, you cross over between non-sister chromatids. Okay? Let's go to metaphase 2 and metaphase 1. Yeah, pause here. Any question about how we make use of all these terms? Okay, but I think they are quite important. This is where I think a confusion will come in. Okay? Okay? Alright, so let's go to metaphase 1. What would metaphase 1 look like? Okay, what would metaphase 1 look like? Okay, present. You go and reposition. What would metaphase 1 look like? Right. Thank you, Lisa. And if I have to look at middle plate will be here. Okay. So if I want to compare, what's the difference between metaphase 1 of meiosis and metaphase of mitosis? Right? What's the biggest difference here? The biggest difference is that when they line up at the midline, the middle plate, they pair up. Right? Or meiosis 1. Okay, sorry, metaphase 1. When we think back to mitosis, there is no pairing up. Just now, Hima helped me arrange, and then we had to slant it to the side. Right, Crystal? Yeah? Crystal and I, we had to slant it to the side, one single file. Right? Compare metaphase and metaphase 1, this is the biggest difference. If there's an essay you like to prepare for this topic, compare and contrast. Right? Mitosis and meiosis. If I were to compare prophase 1 and prophase, what is the biggest difference? The phase we just talked about, prophase 1 and prophase, what's the biggest difference? Prophase 1 got crossing over, prophase 1 that synapses, bivalence form, lots of differences. Yeah? Okay, so prophase 1, metaphase 1, something you can compare there. Therefore, when we go to anaphase 1, what separates are not sister chromatids, but the entire chromosomes themselves. Yeah, we separate entire chromosome. That's another point of comparison. You could compare anaphase 1 to anaphase. In mitosis anaphase, the sister chromatids are pulled apart. But for anaphase 1, it's the homologous chromosomes that are pulled apart. Then let's go to telophase. Telophase, they will all make a single cell. Then we move on to the next phase, right? Okay, so let's now go on to metaphase 2. Anaphase 2, you know, the second part. I think we need to replace this. Yeah. Okay, so now let's go on to the next few phases. What is the difference? Let's go to prophase 2. What would prophase 2 look like? Assuming this is one cell and this is another cell. What would prophase 2 look like? Just a jumbled mess. Just a jumbled mess. What would metaphase 2 look like? Metaphase 2 will look like this. Okay, yeah, got it. What would anaphase 2 look like? Look like this. What would telophase 2 look like? Okay, they start to split up already. Started to change everything. So, meiosis 2 is kind of like mitosis. Right? Meiosis 2 is kind of like mitosis. What you observe. It's just that we are working with half the number of chromosomes. Okay? Okay, maybe to end off, let's talk about what independent assortment of chromosomes is. reverse back to metaphase 1. What is independent assortment of chromosomes? A, B, what is independent assortment of chromosomes? Independent assortment of chromosomes means that how any of these pairs align themselves along the midline is independent from each other. What I mean by that, look at this big A, small a, right? I can put them like that, right? There's nothing stopping me or the cell from arranging itself this way instead, along the midline. Right? It could have been like that, or it could have been arranged like that. How this chromosome here, this pair, arranges itself along the midline, also is up to itself. It could arrange itself this way, right? Big D, small d, or it could arrange itself this way. Small d, big D. How each pair of chromosomes arranges itself is independent of each other. Yeah? That's what independent assortment of chromosome is. But this is important because how they arrange themselves will result in who goes into which cell. Okay? That's independent assortment of chromosomes. It's like me shuffling cards. Okay? Shuffling cards along like that. Okay? So all these terms, the most I find, the part that is a little bit confusing for meiosis is just the crossing over portion to me. Right? For me, how I visualize this better is if I draw everything out. Drawing helps me visualize every step along the way. Okay? The next lesson we come in, we will play the couple one more time. Right? Just to recap. But you play as a group or as a pair this time. Okay, you can discuss with each other. Right? I will go and check if the textbooks are ready. If the textbooks are ready, next you can also read out a bit, and the next lesson we'll start to go into reproduction. Okay? Your timetable changed a lot. So nothing changed. So is it physics after this? Okay, you don't go collaborate with school now.
I'm not.